guys, I'm really stressed. I have so many coordinates left. I just don't know what to do anymore.
I know, I'm scared I'm gonna fail the class.
I know, I think I have like 12 sections left.
Really? I haven't done any of them.
Oh my god.
Oh my god, that's like really bad.
No.
That's just,
What's that?
Oh, I think we have a biotext next week.
We do.
What?
Oh my god.
What?
Oh my god, yeah, we do.
Guys, I think my friend sent me a studying link.
This is a DNA strand. It contains all our genetic information, from blood type to eye color. How does this, an anti-parallel double helix of nucleic acids, actually make us who we are? Today we'll be covering the steps of this vital process. This is transcription. Now to introduce our first character, transcription factors. These transcription factors bind to the TATA box on the promoter region, helping the RNA polymerase bind. Here it comes, along with even more transcription factors. Altogether, they form the transcription initiation complex. Before they begin creating a sequence of mRNA, a segment of the DNA, the enhancer region, folds over and touches the complex to get it going. Now to zoom in. RNA polymerase is separating the two strands. There's a template strand and a coding strand. The template is used to make the new strand of mRNA, and the coding strand has an identical genetic code to the mRNA, other than having uracil instead of thymine. The molecular structures are also very similar, except RNA has ribose sugar instead of deoxyribose sugar and is single-stranded instead of double-stranded. Elongation is when polymerase is creating the mRNA sequence by matching the base pairs from a template strand. The new strand of mRNA is built in the 5' to 3' direction. In this new strand, adenine is being matched with uracil, thymine is being matched with adenine, and guanine is being matched with cytosine. It appears our polymerase has reached the terminator sequence. It now separates and can head home with the satisfaction of a job well done. Don't worry, our adventure isn't over just yet. This is only the pre-RNA. Before coming into the world of cytoplasm, we must make some modifications. A 5' or GTP cap is added to the 5' end, and a poly A tail is added to the 3' end. Still, we aren't done. Enter the spliceosome. See? One strand of mRNA can actually produce multiple different proteins. How? Through cutting out specific portions of the DNA and keeping others create different combinations. Together, multiple SNRPs create a spliceosome. The spliceosome cuts out multiple intron segments, and the remaining pieces, which will eventually be expressed into proteins, are exons. Our mRNA is now ready to head out through a nuclear pore to be translated. Bye-bye! But wait, what if a mistake was made when pre-mRNA was being transcribed? These are called mutations. There are two main types of mutations, the frameshift and point mutations, with respective submutations for each. Frameshift mutations are when one or more nucleotides are inserted or deleted, affecting the entire sequence afterwards. Point mutations are the replacement of a single nucleotide. They can be either silent, missense, or nonsense. Shown on screen is the silent mutation. This means that despite a change in the nucleotide, there will be no effect on the amino acid expressed. According to this codon chart, CCA and CCC will both result in proline being expressed.
Hey guys, Kylie! It's time to rain more heavily.
Wow, guys.
I feel so ready.
Honestly, I think we're gonna pass.
I think so.
Straight A's?
So glad my friend sent me this.
For sure.